Welcome to another day of Mr. Mann's Anatomy and Physiology class. Hope you guys enjoy. So today we're going to talk about the muscular system. We're going to do part one today, and we will do part two the later part of this week. So what we're going to do is do overview of muscle tissues. I'm looking at skeletal muscle specifically within the muscular system. So in the skeletal muscle, we have some terminology, some prefixes. So we have myo and mice, that is going to mean muscle, and sarco means flesh. And then it's going to contain multinucleated cells, which means it has multiple nuclei within each cell, and is striated, visible bands / stripes. And it's under voluntary or conscious control, which means if something's coming at you, you're going to react in some form or fashion to contract those muscles to move out of the way or to catch it. So some connective tissue wrappings and coverings called fascia. And so we have endomyosin, which is going to cover each muscle fiber. We have our fascicle, which is going to cover the bundle of muscle fibers. We have the perimysium, which covers each fascicle, and we have the epimysium, which covers the entire muscle. So if we're looking from deep to superficial, we're going to go endomyosin to perimysium to epimysium. So the epimysium is going to blend into either a cord-like tendon, which is that dense regular connective tissue we talked about previously, or a sheet-like aponeurosis, which attaches muscles indirectly. So we either have it dense regular connective tissue or sheet-like tissue. They both make into the tendon. And so here we go. Here's some labeling for you. We have our endomyosin, which covers the muscle fiber, the fascicle, which is a bunch of muscle fibers. The perimysium is going to cover the fascicle, and the epimysium is going to cover the muscles. So then here's our tendon that attaches to the bone. And that's how muscles are attached, is through tendons and ligaments. So muscle functions, we're going to be producing movement here. So skeletal system, or skeletal muscles are responsible for locomotion or that movement of the skeletal system and facial expressions. Smooth muscle and vesicles and cardiac muscle work together to circulate blood, maintain blood pressure. And then smooth muscles of hollow organs force fluids, in a baby through body channels. It's also going to help maintain posture. Skeletal systems or skeletal muscle works continuously to maintain a seated or standing position despite the downward pull of gravity. So this is why our muscles are very strong. This is also why it's very, very important for as an infant to do your tummy time and to be able to crawl and stand and those types of things to be able to help these muscles go against gravity. Stabilizing joints. Skeletal muscles stabilize and reinforce joints that have poorly fitting articular surfaces. So for instance, our shoulders and hips are very, very important here within using muscles to stabilize these joints. We also can generate heat. So heat is a byproduct of muscle contraction. 3/4 of energy from ATP escape as heat. And heat is necessary for homeostasis and maintaining body temperature. So microscopic skeletal anatomy. Muscle cells are called muscle fibers because of their structure, length, parallel, wrapped in bundles. We have our sarclema, which is specialized plasma membrane. And then make up that sarclema, we have myofibril, which is long organelles inside of the muscle cell. Then we have our sarcomere, which are contractile units of the myofibril. And then sarcomeres are made up of myofilaments, or thread-like proteins. We have a thin and thick filament, actin and myosin. And this is actually a junction here, and this is a whole complex that we'll talk about. Then we have our sarcoplasmic reticulum. It's going to specialize smooth endoplasmic reticulum, surrounds each myofibril, and it's going to store and release calcium needed for the muscle contraction. So here we have muscles to fascicles to muscle fibers to myofibrils to thin and thick filaments, or our myosin and actin filaments. We have our bone, we have our tendon that attaches to the muscle. we have our fascia, and then all these that you need to label as you go through. So I would pause this and label these. Then we have our segment of muscle cell. So again, some more labeling for you. And so we're going to look specifically at a myofibril here of the and then look at the sarcomere of a myofibril. And so these thin blue structures, okay, is going to be our actin, and the thick red is going to be our myosin. In this case, again, this blue is our actin, the red is our myosin. And then in between, when we pull these together, it's going to create what we call an M line for allowing for contraction, which we'll look at here in a second. So muscle contraction. So neurons stimulate skeletal muscle fibers. So to contract skeletal muscle cells, it must be stimulated on nerve impulse. We have a motor unit, or one neuron, and all skeletal muscles that it stimulates. So basically, if I take this picture below here, right here, this neuron and all the muscles that it stimulates would be a motor unit. And so, depending on what type of activities, you might recruit more motor units to be able to do that if it's a very heavy or extraneous exercise. Neuromuscular junction is where neurons meet a muscle cell, as we see here. Then we have synaptic cleft, a gap between a neuron and a muscle cell, also called a synapse, which we'll look at neurons here in a second within this. So this would be our synaptic cleft here, right there, or what we call a synapsis. And I would label synapsis right here, or right here, either one. Okay, so we have some steps in a muscle contraction. So an action potential down the axon triggers the release of a neurotransmitter or some type of chemical that says we need to do something. And it's going to be called acetylcholine into the synaptic cleft that causes action potential or this electrical signal into the muscle fiber. Okay, the action travels along the sarcolemma and inside the muscle fiber to the sarcoplastic reticulum, and the SR is going to release that calcium. Calcium ions are going to be triggered by the binding of myosin to actin. Then we have our cross-bridge cycling occur and the sliding of actin across the myosin to cause the muscle to shorten and thicken along that M line. And so then here's our steps here. I'm going to zoom out just a little bit. So we can see step 1 here. The myosin cross bridge is attached to retain the microfilament. In step 2, the working stroke of the myosin head pivots and bends and pulls on the actin filament, sliding it towards the M line. So we see here, we're actually using ATP here. Is being released. As the new ATP attaches to the myosin head, the cross bridges detaches. And then as ATP splits into ADP and phosphate, cocking the myosin head occurs. So being able to cock the head like this is we're going to release ADP and phosphate. We're going to add ATP for the myosin head to be released, and then we're going to get rid of ATP again to get in position for the next turn. So now what I want you to do is I want you to fill out the next the steps, the 4 steps through muscle contraction. And then last but not least, we have energy for muscle contractions. We have ATP, or adenosine triphosphate, and is needed to power the contraction in only a few seconds where the ATP is stored in muscles, after which other pathways produce ATP. So we have creatine phosphate, CP, which is a high energy molecule, and CP interacts with ADP to create one ATP molecule, and all the supplies for creatine phosphate are exhausted in less than about 15 seconds. So as we see here, creatine phosphate plus ADP yields ATP in a chemical reaction. We also have aerobic respiration. This is the one you remember from cellular respiration. We have glucose. It's broken down into carbon dioxide. CO2 and water are released. And we make about 36 ATP molecules per 1 glucose. And this requires a continuous supply of oxygen. And if we don't have that oxygen, then we're going to go into anaerobic respiration or lactic acid fermentation. And this is what happens when we break down glucose without oxygen. And so this is for those long exercises where you start to feel a lot of muscle soreness here. And so we have 2 ATP molecules per 1 glucose, and the byproduct is lactic acid, which is going to promote muscle fatigue and soreness. So that's going to be it for our notes today. Make sure that you copy these down, turn these in before 11:59 p.m., and be ready to kind of go over this on Tuesday/Wednesday in your class period, because we're going to talk about it. I'm asking some questions to make sure you understand it, and then we're going to get into some micro-macro labeling. Other than that, you guys have a great day, and I'll see you next time.